Hi, welcome. It's nice to see all of you today, even if it's virtually. So my topic for today is immunomodulators, and I'm subtitling it Biologics for the Treatment of Food Allergy. So just to begin, why would we consider biologics for the treatment of food allergy? And I think there are a number of potential advantages that could be very meaningful. The first of them is that they are not likely to be food specific. And while there's been a huge amount of emphasis on food-specific treatments for food allergy, the reality is that many, if not most of our patients have multiple food allergies. And the appeal then of a food-specific treatment, say a peanut-specific treatment, for the patient that is allergic to 7 other things may be far less than it might be for the patient that turns out to be not that common that only has peanut allergy. A second advantage is intermittent dosing, as opposed to the burden of daily dosing that we're talking about with many of the treatment approaches. This is something that would be more akin to an allergy shot, or more akin to getting a shot of Amlisumab for your asthma, getting a shot every 2 or 4 weeks without having the need for daily dosing. A third potential advantage is safety. And so these products overall have developed rather reassuring safety profiles. And compared to the risk that we may be seeing with some other therapies, there may be a great deal of reassurance for patients in terms of, will this treatment for my peanut allergy put me at risk for anaphylaxis? Another that is kind of really neat and really appealing to me is control of comorbidities. And just like most of our patients don't just have a single food allergy, very few of them have just a single allergic disease. So we're looking at patients who typically have more than one allergic disease. And along with food allergy, whether it be eczema, asthma, allergic rhinitis, or some combination of those, these different drugs may have different profiles for treating the comorbidities that could be very appealing to one patient or another. There's a question mark here about disease modifying. I'm not as optimistic about this. I think that controlling disease is something we do rather nicely in our specialty. Modifying disease, whether it be treating asthma, allergic rhinitis, has been rather difficult. But there is a chance by their different mechanisms that there might actually be a disease modifying effect. Now, the biggest disadvantage as I see it is that these treatments will be required for long-term use. As we do with our use of biologic for asthma or atopic dermatitis, we are not curing the disease. We are providing control of the disease that will only remain with regular dosing. Now, while you might think that sounds kind of bad, I don't want to be on this for years and years. On the other hand, most of the other treatments we're talking about are going to be used for years and years, and we don't have any plan, say, for oral immunotherapy that the majority of patients will be able to stop it at some point and maintain any of the protection that the treatment provided. So there are a few biologics that have been under study for food allergy. Omelismab may be as monotherapy or as adjunctive therapy, meaning using Omelismab along with some other approach, such as oral immunotherapy. Dupilumab is now being studied. It's being studied both as an adjunctive therapy to peanut OIT and as monotherapy for peanut allergy. Very exciting to see each of these products being studied for food allergy. I won't be focusing on this, even though I'm very excited about it, because we don't have any results yet from these studies that are now ongoing. Anti-IL-33 has now had a proof of concept study completed with some encouraging results. So hopefully this will make it back on the pipeline to move into a larger phase 2 trial with, again, some pretty encouraging initial proof of concept results. Now, there's a large number of biologics of interest, and they range across a bunch of different pathways. And one of those is Omelismab, which is a different anti-Ig molecule. It may have some advantages over Omelismab, may not have advantages. These are all going to be under study. Is under extensive trial right now for urticaria with encouraging results, and hopefully will be seen in the world of food allergy in the not too distant future. I will say that anything I say will be only publicly available information. So while you and I might know about some studies that are planned or even almost underway, I think I've captured everything that is truly under study versus those that are of interest. Next biologic of interest is the anti-TSLP product, now under trial for asthma and some other allergic diseases. There are BTK inhibitors that are under study for a wide range of diseases, but ibrutinib has now been published as a potential therapy for preventing anaphylaxis, and hopefully, again, will enter the world of food allergy investigation. Anti-SLGLT8, anti-IL-5, anti-IL-5 receptor, anti-IL-13, anti-IL-9 are all products with the potential to impact food allergy. They may be more upstream or more downstream, but again, my personal view on this is that I would love to see the more products studied, because we're not going to learn about the true potential benefits unless they are studied. And we're actually going to learn a lot about mechanisms of food allergy treatment, and how these products prove out in the long run because of the specific pathways that they are impacting. As I mentioned, I'm going to focus on Omelismab, which is a product with a modest amount of data for food allergy. I'll show you a little bit of the data and then dive into a study that we're currently doing in a relatively large scale, looking at the potential for Omelismab as monotherapy for food allergy. And the mechanism is well known to all of you, so I'm not going to go into any detail on this slide. But again, when we think about upstream, downstream, this is way downstream, and maybe way downstream, we're actually blocking the IgE that induces a reaction is going to be most efficacious. Again, we won't know that until the other products have been explored, but there is a great rationale behind why an anti-IgE approach could be successful to treat food allergy. Just to show a few different studies and with some background, and this is a study that we all became familiar with a long time ago. Remarkably, we're almost 2 decades out from when this study was published on the product TNX-901, analog of Amlisumab. And this study was published in 2003, showing a very clear dose-related effect on peanut reactivity. So looking on the left-hand side of the slide, you see that as in many of the studies, there is some placebo effect. But when you go over to the highest dose used, 450 milligrams of TNX-901, there was a highly significant increase in the threshold required to cause an allergic reaction. And this study, for very good reasons, led to lots of enthusiasm about moving forward with anti-IgE for the treatment of food allergy. But for a variety of reasons that are outside the scope of this presentation, things got stalled for a while. And some of those were, specific to concerns about the studies that might be required to get a product license. Some of those were the product itself. So TNX-901 was taken off the market, and studies were proposed to move forward with Omelismab, which got initiated, but unfortunately were stopped relatively quickly, not because of problems with the drug or problems with the results, but problems with adverse reactions to the food challenges that were needed to proceed with the full therapeutic trial. So we took a look a number of years ago now at Omelismab as a very straightforward study here for the treatment of adults with peanut allergy. And what we did in this study is there was a baseline peanut oral food challenge. We then did weekly blood draws to assess bronchial hyperreactivity. And we then re-challenged once the basophil hyperreactivity was reduced by 80%, but no later than week 8. So subjects all had received between 1 and 3 doses of Omelismab at the time of their second food challenge. Then all subjects continued on the Omelismab a full 6 months of treatment before their final food challenge. And these are the summary results here. What we're looking at here is the change in peanut allergen threshold to induce an objective response on food challenge. And in the blue lines way on the left, you'll see that there are 6 subjects after getting just 1 or 2 or 3 doses of Omelismab who went from being highly allergic, reacting to less than 100 or 200 milligrams of peanut protein, to tolerating 10,000 milligrams at their first food challenge. And then the group sort of divided out into a subset of really great responders and the subset of less great responders. And we almost consider them non-responders over 4 lines on the bottom of the graph. But when you really think about it, when you compare this degree of improvement to other therapies under discussion, getting to 2,000 milligrams of peanut is really quite protective. and certainly as good as anything else that is being investigated right now. When we break it down a little bit further, we again get this sense of the great responders and the not great responders. And very interestingly, if you look at the response, it pretty much divided up in that very first food challenge, again, all within 8 weeks of initiating the Omelismab, 1 to 3 doses in total. And additional improvement was very unlikely to be seen in this small study after getting an additional 4 or 5 months of Omelismab injections. So it's sort of interesting to me that there does appear to be a range of response. There are some people with dramatically good responses, some people with lesser responses, although they are still gaining apparently some degree of protection, if not a major degree of protection. And they declare themselves fairly quickly as to those who are going to be the great responders and those who are going to be less good responders. Now, other studies, we have to extrapolate. We don't have direct challenge thresholds, but there have been a number of studies now looking at Amlisumab as an adjunctive therapy to oral immunotherapy. And in this study, looking at peanut OIT, with or without Amlisumab, it was shown that if you had the pre-treatment with Amlisumab, on the left-hand side of this graph, you had a dramatically greater chance of hitting the maximum allowed initial dose escalation of 250 milligrams. So on the placebo group, the median was 22.5 milligrams. On the Omelismab group was 250 milligrams. And it might have even been higher if the study had allowed the initial dose escalation to go beyond that 250 milligram mark. And then the last study I'll show you here is another looking at the concomitant use of Omelismab and oral immunotherapy. In this case, though, looking at multi-food immunotherapy and looking at achieving a rather ambitious maintenance dose of 2 grams of each food, and demonstrating here in the red line that those subjects treated with Omelismab had a significantly greater chance of making it to this rather large dose, 2 grams of each food, than those on placebo. And those on placebo, some of them did make it, 35% did get to the total 6 gram of food, but it was clearly different. And another sort of insight into one potential use of Omelismab or other anti-IG approaches for food allergy, again, whether it be as a monotherapy approach or as an adjunctive approach, to either make the other approach, such as oral immunotherapy, safer or much more convenient because you can reach desired maintenance doses so much more quickly. Now, I want to finish the rest of the talk really talking about a study that we are actively involved in right now. It's part of the COFAR network, a consortium for food allergy research. And the title of the study is Omelismab as monotherapy and as adjunct therapy to multi-allergen OIT in food allergic children and adults, but the focus will be on a pediatric population. So there are 10 centers involved. It's double-blind, placebo-controlled, 225 subjects. The age range is 1 to 55 years who are allergic to peanuts. They have to have peanut allergy, and they have to be significantly reactive to at least 2 other foods. And those come from a list of milk, egg, wheat, cashew, hazelnut, or walnut. This is a phase 3 trial. It's a collaborative study between COFAR and Genentech Novartis. And I'd like to just take a second to say how valuable I think this kind of collaboration is. As more of our food allergy treatment investigation has moved to industry, I think that this potential to work between agencies, whether it be COFAR, whether it be other NIH, whether it be FAIR plus pharma, all have great potential to move the field forward more quickly than we could do on either side on our own. And Genentech Novartis received breakthrough therapy designation from the FDA for Omelismab for the treatment of food allergy. So this is a complicated study, too complicated to review on this schematic, but to mention that there's a fairly intense screening period where each subject has to prove reactivity to these 3 foods to which they will be treated. The reactive dose for peanut is a cumulative dose of 144, and for the other foods, it's a cumulative dose of 444. And once you have achieved that, you've met all the other inclusion and didn't meet any other exclusion criteria, you then have the opportunity to undergo these baseline food challenges with 3 active challenges and 1 placebo. Then there's a stage 1, a stage 2, and a stage 3 that's probably easier to look at in a text format. So stage 1 is to evaluate the effects of Omelismab, here using 16 weeks of treatment as monotherapy compared to placebo. The primary endpoint is based on that peanut reactivity with a consumption of greater than or equal to 600 milligrams, cumulative dose of 1044 peanut protein during the end of stage 1 food challenge. There are many other secondary endpoints, including the other foods, which we've set the bar a little bit higher because their entry food challenge criteria was a little bit higher. At greater than or equal to 1,000 milligrams of cashew, milk, and or egg, we focused on those 3 because we thought we would be best powered for those 3 additional foods. And other secondary endpoints are looking at reaching higher thresholds. So we're not just going to stop the challenge because they got to 600. They're actually going up to 6,044 milligrams in the end of stage 1 challenge. So we will know the degree of protection far beyond what we're investigating as the primary endpoint. Stage 1 also includes an open label extension for the first 60 subjects treated with Omelismab, and that was requested by the FDA to study the durability of longer term treatment. And all stages have extensive mechanistic endpoints that we're very excited about to be able to learn more about what's really going on, especially as we start to get into this, good responder, less good responder profile that we might end up seeing based on our preliminary studies. Now, stage 2 moves to secondary aims, and this is comparing treatment of patients with multi-food allergy using longer-term Omelismab as monotherapy or treatment with multi-allergen OIT using Omelismab as an adjunctive treatment. And the adjunctive treatment will be giving Omelismab 8 weeks before and during the first 8 weeks of OIT. And this will remain placebo-controlled. So those getting active Omelismab through the entire 52 weeks of stage 2 will be getting placebo OIT. And those getting active OIT will be getting placebo injections once they complete this 16 weeks of pre and slightly into treatment with open label Omelismab. Then stage 3 is something that I'm really excited about because I think it can really help to move the field forward. And our goal of stage 3 is that once you have completed stage 2, we will know what degree of protection you have, whether it be because of the Omelismab or because of the OIT, and we will then move to introduce foods in a very regimented dietary form into the diet. Now, they could get rescue OIT if they need it, but the goal really is to make this less medicinal and to move patients into a real-world setting of being able to begin to eat the food. And we're even hoping that on Omelismab side, we will be seeing patients becoming rapidly desensitized. So they finish their Omelismab, they introduce the food, and we're hoping that under that protection of Omelismab, as it wanes in their system over the next 2 or 3 months, they'll be able to be eating the food in a sufficient quantity to become desensitized. So we think the study is going to provide groundbreaking data that will impact the future of treatment for both peanut allergy and multi-food allergy. And we'll also have the ability to study the safety and efficacy of these treatments down the age of 1 year. We initially proposed this as down to age 2, but we've now been able to expand it down to age 1, which we're very excited about. Now, the study status is ongoing. Enrollment is now between 20 and 25%. We were at that point basically in March when COVID hit. COVID was a blow to all of our research. I think it's actually worth noting what a disruption that has caused to all research, whether it be food allergy or otherwise. And all of the subjects at the point that the study was halted who had been randomized were either in stage 1 or the open label extension, meaning they were getting Omelismab or placebo injections, and they all had to go back and restart. So if you'd already been on 12 or 14 weeks of injections out of the 16 weeks of stage 1, you had to go back and start from the beginning. We did not make anyone restart their food challenges, but it was sort of a very big imposition on these families. And so far, we have 100% of people volunteer, actually very enthusiastically, to come back and restart their stage 1 or open label extension injections. Expected results, if we do well, we will complete recruitment in the next year. Again, there's 16 weeks once everyone is randomized. There's then the data analysis, but ideally in the next 18 to 24 months, we will have results of the primary endpoint, which we are hoping will lead to a label for Omelismab for the treatment of food allergy. And lastly, what's the future biologic for the treatment of food allergy? I think it's unlimited. It may fall to the wayside because other treatments are so much more effective. But I think the advantages that I showed in the first slide about not being restricted to a single food, treating comorbidities, having safety, not having to do a daily dose, all make these drugs quite appealing. And for unusual food allergies, so I have patients whose lives are restricted by their peanut allergy, but they're far more restricted by their milk and egg allergy. And if we could do something to allow them to have less restricted lives, and hopefully, as we're going to look at in stage 3, even begin to introduce the food into their diet, or those patients with very unusual food allergies, patients whose lives are completely limited because they have a severe allergy to something like mustard. I don't think we're going to be providing effective immunotherapy to mustard in the foreseeable future. And for that patient to be able to get a shot every 2 to 4 weeks and be protected, be able to go out to a restaurant and eat safely would be a fantastic opportunity. So I look forward to discussing all these opportunities further as we move through this exciting summit and as we complete these studies over the foreseeable future. Thanks very much.